Okay, we should be live. Yeah, so we'll give it about 1 minute and then we will get started just to let people come onto the stream.
Good.
Okay. So hello everyone and welcome to the Paper Trail. My name is Bria Macklin and I am a postdoc at the Gladstone Institutes. And so the Paper Trail is an online interactive journal club that was developed by 4 former colleagues turned lifelong friends. So myself, Michael Blatchley, who is currently a postdoctoral fellow at CU Boulder, Franklin Hall, who's a graduate student at Johns Hopkins University, and he's going to be your host today. Michael Blatchley, I am Quentin Smith, who is an assistant professor at UC Irvine. So born from our love of discussing science and our yearly paper a day challenge, we came up with the idea for the Paper Trail to be a platform that allows primary authors to come and share the story of their research. And so we really wanted the Paper Trail to stand out from other online presentations. And to do this, we came up with a set of guiding questions that prompt the author or authors to share more than just their results. And so we're so happy that you've decided to join us for this live stream. And in true Twitch form, we ask that you use the chat feature to ask questions throughout the presentation. So the other hosts are serving as moderators in the chat room, and they'll relay any questions that you have to Jeremy and Eric, who are eager to answer your questions throughout the presentations. So please don't hold them to the end, ask them as they kind of pop into your head. And if you don't currently have access to the chat feature, what you can do right now really quick is just create a Twitch login and then come right back and you should have access to the chat feature. And so with that, I would like to introduce you to our speakers, Dr. Jeremy Huang and Aric Lu. So Jeremy is a postdoctoral associate at MIT under the guidance of Professors Linda Griffith and Douglas Laufenberger. His aim is to study the mechanisms behind disrupted tissue immune interactions in uterine diseases. And in the future, he hopes to become an independent investigator within a bioengineering department. And so Aric is a 4th-year PhD student in Jennifer Lewis's lab at Harvard University. His research focuses on using 3D printing and genetic engineering technologies to create multicellular tissues. And outside of the lab, he enjoys running, cooking, and streaming on Twitch occasionally. And so we'll drop his Twitch username, his Twitch channel in the chat if you guys want to check out his Twitch stream. And so with that, I'll turn it over to Franklin and you guys can share your slides. And yeah, really excited for this presentation.
Yeah, thank you, Bria, for the kind introduction for everybody. And yeah, when you guys want to share, you can take it away.
Yeah, let's go here actually. And now let's see. Yes, hello. Okay. All right, we all good?
Yeah, looks great.
Let me minimize this window here. All right, Jeremy, you want to start?
Yeah, thank you for the kind introduction, Bria. So the talk today will be on our paper, which was recently published in Nature Biomedical Engineering, entitled Orthogonally Induced Differentiation of Stem Cells for the Programmatic Patterning of Vascularized Organoids and Bioprinted Tissues. And obviously, such a work cannot be done without a multi-talented and multi-disciplined team. Especially, I want to shout out Dr. Mark Schuyler Scott, who's now a professor at Stanford University, and Tomeo Dunkel, Lucy, Sarita, Alex Ng, Songlei Liu, and the corresponding author, Dr. Jennifer Lewis, and also my old boss, George Church, who are on the who's on the paper, and our funding sources, NIH, NHGRI, and the Office of Naval Research. So with that, prior to the COVID-19 pandemic, there was another scare there. And if you remember, back in 2015, there was a great concern, in particular of the Zika virus, which is transmitted by mosquitoes. And really really the primary phenotype was that babies were being born with stunted brain development, right, with severe microcephaly. And there was a great call to scientists, particularly at that time, to understand how what's the mechanism behind the mechanism action of Zika virus, in particular, to cause this phenotype. And fortuitously, people have been working in the modeling in the in vitro engineering space to create models of the brain. So this was a very simple experiment published in Science, where they created brain organoids and cultured them. And then they introduced Zika virus and showed that Zika virus actually targets the neural stem population. And how are these models created? Well, back in 2013, Madeline Lancaster and in Nobelich's lab had created a protocol where you can create these brain organoids from aggregates of human pluripotent stem cells, where you would aggregate it, and then through successive media differentiation method introductions, you can create a neural induction of an embryo body and then embed it into an ECM like Nature Gel. And these would create little neural rosettes, and over successive media introductions, you can differentiate them into more mature cells. And this process is not just in the brain, and for researchers all over the world have been using pluripotent stem cells to make these kinds of tissues in not just the brain, but for example, cardiomyocytes, adipocytes in the bottom left. But just to highlight Madeline's initial paper in 2013. But you know, in the 8 or 9 or so years since Lancaster created these tissues, teams across the world have been interested in creating more complicated, more biomimetic engineered tissues. And because brains are layered and diverse and, for example, vascularized. And so Madeline utilized chemicals, growth factors, and to stimulate pluripotent stem cell growth and development. However, this particular process is not very specific, and it's akin to rolling a ball down a slope. You will, if you're guiding it, you would probably get, for example, to a Neuroectoderm lineage, and this is a picture of Waddington's famous epigenetic landscape. But we were curious to ask the question: can you actually get cells of specific identity, for example, and specific layers, because the brain is actually highly structured, and the organoid that you see on the left is not terrifically structured. There is structure there, and the cells that are there are not specifically patterned or not specified. So we were wondering, can we actually create complex models from cells from vastly different lineages for drug development assays or future neurodevelopment assays? For example, if you were to ask the question, can a drug cross the blood-brain barrier and develop its payload, you would have to create a tissue that has capillaries and pericyte supporting cells and astrocytes, for example. So again, how do we create these complex models from cells of different lineages? And we're really hoping to create these kind of vascularized perfusible structures in the future. So a lot of people in the field have been mixing different cell types and engineering cell types to achieve this aim. And the reason, and there's a lot of ways to direct the differentiation of cells. For example, as a review, I would like to think of there's 3 primary ways. People modulate surfaces, right? Cells have integrins or receptors that are responsive. And the aforementioned chemical ways where you can add cytokines or growth factors will also push differentiation. But we were particularly interested in using genetic engineering to directly regulate or directly influence gene regulation in the nucleus. And a lot of people have been doing this within this space, and we were interested because it particularly in the Church lab, and we had Volker Buskamp back in 2014 had demonstrated rapid differentiation of human pluripotent stem cells into mature neurons within 4 days. So he used a combination of 2 transcription factors, neurogenin 1 and neurogenin 2, in a cassette. And this cassette is doxycycline inducible with a reverse tetracycline activator. And so I like to think of it as akin to, if you have, I love music, and if you're like a mixer producer, you're just kind of like ramming up a signal to the max, right? And you're trying to see, okay, what kind of signal do you get there? And this motivated, in particular, our colleague Mark Schuyler-Scott to talk to Dr. Alex Ng, which created there's a question in the chat.
Yeah, so that's a pretty quick differentiation through that transcription factor kind of cassette you just showed us. How does that compare to what you would do normally with media kind of differentiations in terms of time length?
That's a great question. And up till this point, especially transcription factor differentiation, like I said, it was rapid within 4 days. Media typically to get mature neurons on the dozens of days order. And also, as something to note, the purity of this population was extremely high and validated. And if you're interested, you can go into their original paper where they have RNA sequencing data to validate this. But if you were to use the media methods, the purity is across the board, but also the speed is much slower, like dozens, 30 days plus.
Is there any kind of like initial trade-off that you guys know of yet doing transcription-based versus media-based differentiation for these, or is that really, not really exist yet that anybody's seen?
Well, you are inserting for example, you are inserting a cassette into the genome. In this case, there was so it could knock out if methods since then have tried to find safe harbor sites to insert it and then withdraw it. For example, if you want to in the future create these for stem cell therapies, you don't want to be inserting a cassette and leaving it there if you want to put it into a patient. So that's one negative thing.
Okay.
But you are also specifying a very specific subpopulation of cells, so you don't get cellular diversity. So you can think of it whether or not it's a positive or negative. Mm-hmm. It's also difficult, and I think this is a general difficulty within the field, is that validating exactly what this cell type is. Nowadays, people use single cell sequencing to try to map these engineered cells onto existing primary cell data sets, but sometimes a weakness is that it doesn't, fully map up into known neurons. You can functionally say this is a bipedal neuron, it may fire, but maybe it's not quite mature, right? Maybe it is mature to an extent. Those are some of the drawbacks, but I would say they're more broad stem cell drawbacks than, and it is one way to do differentiation. But thank you.
No, thanks. Yeah, it's great answer, man.
So in 2015, in particular, the Church Lab had been utilizing this technique of transcription factor overexpression to drive rapid differentiation. And Alex Ng and Mark Scott and I came along pretty much right after. We're in conversations of what if we can co-culture cells together in the same pot and utilize transcription factors to differentiate cells really rapidly. And this paper, which Alex Ng did, he actually created an entire transcription factor library, which is the 1,573 transcription factors plus 200 or so isoforms, and did a multiplex screen of single overexpression and found that there are 200 or so transcription factors that cause pluripotent stem cells from different patient or different lines to be able to be cultured in empties, which is a stem cell maintaining media. And so they actually demonstrated at the very end that you can actually co-differentiate cells in the same stem cell maintaining media to mature cell fates. And that was a very interesting thing and led us to ask the question, well, if we can do this in stem cell maintaining media, can we create, mature neurons and also endothelial cells to augment existing organoid, like cerebral organoid, like Lancaster-like protocols to perhaps give some vascular networks, right? And really, yeah, this is the question that was all the way back in 2015. So our first hypothesis was that transcription factor overexpression is sufficient to differentiate cells from different lineages in neural specifying chemical growth conditions. And this is in particular, just to remind you, for example, Lancaster's method and a lot of the brain organoids method is self-assembly of human pluripotent stem cells and the successive introductions of chemical media, right? And so we were wondering, can we add subpopulations of cells? For example, we call this inducible endothelium or IENDOs, which have a cassette containing ETV2, isoform 2 transcription factor. And then also another subpopulation, what we call iNeurons or inducible neurons, which have NGN1 that we can overexpress on command, right? And so just explanation in using IF stain, if you were to have the stem cells, the non-engineered cell populations and you add doxycycline, they remain as neuroprogenitor cells, right? Nothing really happens. However, if the program cells, if you add doxycycline, right, then it becomes a strongly an endothelial cell population, right, with the staining of the coherent and von Brunnel benefactor on the right. And we also demonstrated, this is in 2D single culture, that for the iNeurons, they become rapidly within 6 days positive for TIJ1 and have like long axonal projections with the addition. And this was further followed up with flow cytometry, where we show kind of robust differentiation with very high efficiencies, right? In endothelium is almost 100%. With our iNeurons, it was around 75 to 80% plus. And we had to ask really a follow-up question. Do these co-cultures, for example, affect one another? Because cells obviously signal, right? We don't want, like, let's say there are neural stem population that maybe potentially influences our programmed cell population. So we co-cultured, for example, wild type non-engineered cells with our iNeurons and then our iEndos, and did a bulk sequencing analysis after separating these populations after co-culture. Are they affected heavily? And really, if you look at these volcano plots, they actually aren't very much. So showing that you can actually pre-program these cells to a particular, for example, lineage, and hopefully introduce some kind of functionality or addition to our organoids. We're thinking about organoids at this time, but this is in 2D. And we actually demonstrated this not just in 2D assays, but on 2.5. Yeah, there's a question.
Yeah, it's curious. As these cells are differentiating into these separate populations, I guess you would assume that they're kind of secreting some level of paracrine and autocrine factors. So to not really see any difference on how that may guide their differentiation is one thing. Do you guys foresee any differences in functionality with co-differentiation versus differentiating them separately?
Yeah, this actually goes into one of the questions, this very astute question, and I believe a reviewer question as well.
Yeah, a little bit.
Yeah, and we did, and we'll talk a little bit about how we tried to look at this in some of our single cell sequencing data, but we were surprised that there wasn't a heavy skew, maybe potentially because it's a rapid differentiation, right? But we had hoped that for example, if the inducible endotheliums were co-cultured, maybe it gains additional kind of neural kind of vascular identity, but it wasn't statistically strong enough for us to claim any kind of additional, it wasn't a strong enough argument, at least in terms of the data in the single cell and bulk seq, to be able to say, okay, there's a strong difference. And we think of it as a potentially as a benefit, right? Because you're programming in, you have in your mind like a cell type of interest and some kind of structure or some kind of introduction that you want to introduce, and you can add them in specifically, right?
Yeah, no, I definitely see the benefit of them not do that. So I could definitely see how you guys use that to your advantage. But yeah, thanks, great answer.
And then finally, we before Aric gets to talk, we also validated this not just in 2D plate culture, but on 2.5D, where we laid kind of a matrix gel layer on the bottom and we co-cultured cells, and we can show we can titrate the different concentrations of our initial mix to create, for example, on the top, combinations of non-engineered cells, which will form these kind of little rosettes, and then adding in different concentrations of inducible endothelium, or in the middle, you would see a combination of inducible endothelium and inducible neurons, and the bottom kind of inducible neurons and in our wild type non-engineered cells. And you can also combine them all in thirds. And this is kind of like a video where I took the GFP non-engineered cells and inducible endothelium in the red and forming tubules and little rosettes. So now I'll hand it off to Aric to talk about the transition to 3 dimensions.
So as Jeremy talked about in his introduction earlier, the whole goal, one of the goals of this work was to be able to tune the composition of these organoids, and specifically of these brain organoids. So what I'm going to be talking about next is the work I started doing when I joined the lab in 2018 or so, about using these cells to control organoid composition and showing how they're compatible with existing organoid protocols and how we can use them to also change how cells are distributed throughout an organoid. So the first question I kind of want to address up front is, why do we need this orthogonally induced differentiation approach to add these additional cell types to organoid protocols? Why, for instance, I couldn't use already differentiated endothelial cells or already differentiated neurons and add those into an organoid to do the same thing? And what we found is that when you try to mix these two cell types together, you have this differential adhesion problem where the endothelial cells greatly prefer to cluster with other endothelial cells, and your iPSCs will also cluster with other iPSCs. And so you, in effect, you get this phase separation, which you can kind of see in this figure here, where we have a ball of RFP labeled HUVEX that's entirely separated away from the GFP labeled iPSCs. And so the advantage of using our pre-programmed iPSC lines is that they're both iPSCs, and then mixing them together will result in a cohesive aggregate where you get this homogeneous mixing of the two cell types without worrying about this phase separation problem. And that's what really kind of is driving this project forward, is this ability to introduce these cell types after any aggregation into a 3D tissue, and have them evenly distributed throughout the tissue and not have to worry about trying to get these cells from the outside in, for instance. And so, like Jeremy had shown in the 2.5D assays, where we're mixing these two cell types, these multiple cell types together, we can do the same in 3D, where we can start all the way from 100% endo cells to all the way to 100% wild type iPSCs and every proportion in between, and we still get a nice mixing of these two cell types. So now taking these aggregates and starting to differentiate them towards brain organoids, what we see is that by introducing these inducible endothelial cells, we're able to force the we're able to introduce this endothelial cell population that doesn't natively arise in brain organoid differentiation, as you can see in our purely 100% wild type organoids versus our mixed 2-to-1 ratio of wild type to eye endocells. And we can see that our endothelium uniquely arises from our eye endocell population. Oh. Question?
Yeah, if you can go back one slide. Yeah, these look beautiful, and the stark difference between having the adult cell population, try to put them together and not getting the mixing versus the other one is it's really different. So it looks beautiful. One question that we had was, do you know why, like what kind of coherence or what could be involved in the cell-to-cell contact that works for the different co-differentiated cells versus the adult cells?
So I think you're right that it is these cadherins, these adhesion proteins that are differentially expressed between the different cell types. I don't know exactly which ones are differentially expressed, but there is some work out of, I think, Magdalena Zernicka-Getz group, where they were trying to solve a similar issue with some of their early developmental studies and using iPSC-derived germ layer cells and getting them to stick together has a similar problem. And so I think what they found is forcing a expression of these cadherins help them get cohesive aggregation. So I think, yeah, you're exactly right that it's the cadherins that are responsible for this behavior.
Okay, perfect. Yeah, these look great.
Yeah. So with these organoids, now that we have this vasculature, we can see that from day 10 to day 25 to day 45 of culture, we have this persistent vasculature that grows and develops alongside the organoid. And Jeremy already showed a video earlier of a Z-stack of one of these organoids where we have vasculature that's intermixed throughout the entirety of the organoid. And furthermore, we see that in these organoids, we preserve these ventricular structures that are characteristic of brain organoids, where we have this nice luminal structure in the middle here, surrounded by a layer of neural stem cells marked by SOX2 or PAC6, and then further away we get neurons labeled by TUJ1. And so we get some of this stratified layering. And importantly, this is all done in brain organoid differentiation media. We don't make any major concessions to the media conditions other than adding a little bit of vascular endothelial growth factor. But by and large, it's all just the same cortical organoid differentiation protocol that's been used by Sergio Pasca or Madeline Lancaster. And we're getting these cells to grow in very non-specific media conditions, which is really exciting for using this in other organoid systems. So now to talk about kind of organizing these, spatially organizing these organoids. So in the examples I've shown before, where we have this random mixture of our cell types, if we want to mix all three of our iPSC, our wild type iPSCs, our eye neurons, and our eye endo cell lines together, we'll get this random mixture of all three cell types, which isn't necessarily physiologically, it doesn't really match the natural physiology of the human brain. So one thing we want to know is, can we template the stratified structure of native brain development in organoids? And so we can do this using, and to kind of show the differences in the way organoids look versus kind of the native brain looks. What happens in organoids is that while you get this stratified layering a little bit, at some point the growth gets arrested and you don't get the development and this expansion of these upper layers of the cortex. And in particular for the endothelium, which isn't shown in these pictures, the endothelium is actually invading from the outside of the brain inwards. And so one thing we were looking at doing was using our rapidly differentiating neurons to kind of coat the outer layer of neural stem cells. And then outside of that, coat the organoid in a last shell layer of induced endothelium to provide the endothelium that's going to invade the native brain structure, the developing organoid, sorry. So by doing this, what we can see is that the way that we pattern the organoids at the start of the aggregation process informs the way these organoids self-organize as they develop. And so in the random organoid condition, I want to point out this cluster of UN positive neurons, which has somehow found its way into the center of this aggregate, and which is very, it's almost like an ectopic grouping of neurons in the center of this organoid. Whereas in the multi-core shell organoid, we see that the neurons that we believe were introduced are all kind of at the periphery of this neural stem cell regime, in between the neural stem cells and the endothelium. And we also see that in our randomly mixed conditions, we have smaller ventricular structures, these nice luminal structures here, versus our multi-core shell, where these luminal structure, these ventricular structures appear larger. And so one of the questions this opens up is, can we kind of inform organoid development by this patterning method? So to kind of demonstrate how the patterning of these organoids is a little bit more useful, what we sought to do was embed our organoids inside a gel containing endothelium and use this as a way to see if the endothelium that we were adding to our organoids could then participate in vascularization and anastomosis with other gels or other endothelial cells in an exterior environment. And one of the interesting things that happens is that when we look at our randomly patterned organoids versus our multi-core shell organoids, we see that the randomly patterned organoids look a little bit more disorganized around their periphery, where we have a lot of cells all migrating outwards into the gel. The organoids are losing a little bit of its cohesion. And we don't see that in the multi-core shell organoids, where we can clearly see this nice round structure of a tight aggregate. And we see a little bit of cells migrating outwards, but visually it looks a lot less than the randomly patterned organoids. And so when we do immunostaining, what we can see is that we still have this nice core shell, multi-core shell structure preserved in the when these organoids are embedded in these gels with other vasculature, when you have all of the cell migration occurring as the endothelium from the embedded in the organoid starts to interact with the endothelium that's outside of the organoid. So the follow-up question is kind of what we did in 2D is, how do these OID cells now affect the differentiation of the organoid as a whole versus just one stem cell line? And what we found when we did the single cell RNA sequencing was that no matter how we patterned our organoids, we didn't have a significant difference in the proportions of the cell types that we got, or the range of cell types we got. And what we also found is that between our wild type only organoids and our organoids with our inducible endothelium and neurons, we don't see really many differences in the distribution of these cells across the different clusters in the organoid. And now there's a question.
Yeah, just actually on the previous slide, just was there any like troubleshooting? This seems like it was probably a little technically challenging to get the gels right. I don't know if you guys had to add any VEGF to kind of help with the growing of the vasculature. Can you guys just run through any of that?
So the endothelium in the gel is the same induced endothelium that we're using within the organoids. So in that way, it's pretty convenient for us to continue using the same media conditions that we were already using for the organoid. But I will say that this is really just sitting in a gel droplet, and you have a lot of cells migrating within the gel, but there's not a lot of things that are happening outside of it. It's kind of just in a little dish with some media on top and cells migrating within this gel.
So we guys first started this kind of endo differentiation. Was this something, I mean, this seems like something that may have come later. You guys realized like, oh, this is another way that this can be used, or was this something like when you first were creating that endo system, that you at that point didn't know that this would be something that you could kind of ultimately do? Is that kind of clear?
So yeah, so there's an interesting answer to this question, because one of the things we were working on a while ago was actually trying to use the vasculature in the organoid to perfuse these organoids. And I will say that these organoids I'm showing here are not being actively perfused. They're just sitting in the gel. There's not really anything connected to them. We were working on that for a while, and then 2020 happened, and we got kicked out of the lab. And so at that point, we hadn't figured out that part of the project yet. And so when the COVID hit and the lab shut down, we were writing up the paper and we didn't have this sort of stuff inside of it. But it was always kind of like in the back of our minds as something, this would be cool to do. It's technically challenging. We don't have it working yet. And so this is actually something that was added to the paper after the first and second round of revisions to the papers, because now we had gotten back into the lab, and we had kind of known that the reviewers would have been asking about, okay, show us what you can do with these certain cell types. This is something we were looking at doing in the back of our mind. And then, after working on the organoids a little bit more after the labs reopened, we were able to include this in the paper.
Cool. That's awesome. Thanks.
So it's a weird reason for not including it, but the pandemic is always a weird reason for everything.
Yeah,
so,
right.
Yeah, so going back to our single cell RNA sequencing data, what we see is that our endothelial cells are only really present in our random multi-core shell organoid conditions. And you kind of see a few dots on the wild type only.
There's two.
Yeah.
It's actually quantified as two.
Yeah, that is actually just 2 dots of probably about 10 to 20,000 cells in that condition. So a few cells ending up in that cluster is not surprising to us. But for our eye neuron cells, these eye neurons, when we made them, had a barcode on them, so we could just uniquely identify the cells that arose and differentiated from our eye neuron stem cell lines. And what we see is that a majority of our eye neurons cluster with excitatory neurons that derive from the wild type iPSC lines, which gives us an indication that, even though we're adding the cells early, they're not significantly biasing or interacting differently with the rest of the organoid. With all that said about organoids, if we want to look at maybe taking all of our tissue engineering knowledge and applying them to start making larger scale tissues that are centimeters in scale, eventually to potentially build something that you can put in a person, right? A millimeter-sized organoid has a long way to go before it gets to the centimeter scale tissues we'll need for organ repair and replacement. So how do we go from building engineered organoids to engineered organ scale tissues? And the way we can do this is we can use bioprinting. What bioprinting allows us to do is it allows us to print these very high cell density cell filaments and create whatever pattern we want within a tissue and let that grow and mature into something that we could eventually, or at least for the time being, like an engineered tissue you could use for like disease modeling or drug discovery or something like that. And so what we're doing here is we're basically taking a cell pellet when we centrifuge all of our cells to harvest them down, and taking the pure cell pellet and extruding that as an ink for bioprinting. And so if we look at a picture of the process in action, we see that this bioink is being deposited and it's holding its shape on this, in this case, this is a transform membrane that we're printing on. And the nozzle diameter here is 50 microns. And we can see that the majority of our cells survive the printing process. We have some cell death at the very edges of our filament, but what we found is that it's not actually due to any shear stress that occurs at the edge of the filament. What's actually happening is the cells in this very high density filament begin to dry out. And so if you don't encapsulate them in another ECM or something that's going to keep them hydrated, the cells on the outside will begin to dry out and die. And so that's why we actually see lower viability at our slower print speeds, where there is less shear stress than we do at our faster print speeds. And so when we looked at the density of these filaments, we found that the density of these filaments is on the order of 500 million cells per ml, which is already at or exceeding the cell densities of a lot of tissues in the human body. So if we take this third order Hilbert curve that we were printing in the previous slide, and we look at it on day 0, we see that we have a nearly pure population of stem cells marked by OCT4. And depending on the cell line you use to print these patterns, you can get neural progenitor cells, you can get endothelium, or you can get neurons that are derived from the cell lines and that derive from the transcription factor overexpression that was pre-programmed into these cell lines. And we can see that the scale of the scale bar on these images is 1 millimeter. So the whole thing is 3.5 by 3.5 millimeters wide. That generally the large scale patterning that we impose with the bioprinting is preserved over the 4 to 6 days of culture that these cells were cultured over. So now printing these third order Hilbert curves is nice and cute, but how do we create arbitrary patterns of multiple cell types and start incorporating all of these different cell types together with bioprinting? One way we can do it is taking sorry, is there a question?
One question was just how you guys derived coming to using the GelBrin for your on top of after you printed. What's kind of the thought process behind that?
The GelBrin was kind of, so are you asking for the reason why we used the GelBrin?
Yeah, why choose that? Yeah.
The reason why is because it worked in a way that one of the things that we had an issue with in printing these structures was when we went to go encapsulate them in a gel, if you pipetted too aggressively, you would just wash away all of your cells and all of your printed pattern. And so for what we found is that the blend of GelBrin that we used did a really good job of just kind of having this laminar flow pattern and doing it in a robust way that we could pipette it on top of the patterns, encapsulate it, and not lose all of our cells to being washed away and lifted up by the movement of this gel across the top of these cells. So these are all really printed on a transwell. So they're all kind of printed on a 2D surface effectively, and then we're casting a gel on top of it. But because it's cells, basically just cells and media, they're very sensitive to any sort of movement of other fluids on top of them that's going to disrupt the pattern.
This is awesome. All right.
So, okay. So moving towards multicellular tissues with bioprinting, what we can do is we can make 2 syringes and print 2 separate inks into whatever pattern we choose. And in this case, what we're doing is we've printed a line of wild type iPSCs, and then following that, as you see where the nozzle is, we're printing a second layer of iNeurons around it, and we can create multicellular tissues this way by just programming where we want to deposit each of these different cell types. A more clever way to do it is to use multi-material bioprinting nozzles where both of these inks are loaded together and printed at the same time. And what we did in our paper here is we did this sort of laminar co-flow nozzle where we have all 3 of our cell inks all loaded together and extruded in the stripe pattern, where we have wild type cells in the middle, iNeurons on the right, and iEndos on the left. And so when we print these, we can extrude all 3 cell types together at once in whatever pattern we want. And the patterning is maintained by this nozzle. And when we differentiate them in the neural progenitor cell culture conditions, we see that this patterning is preserved and we get all the 3 cell types that we wanted to program in the very beginning. So that brings us towards the end of our presentation. In summary, what we've hoped to have demonstrated is the concept of orthogonally induced differentiation and co-differentiating wild type iPSCs with transcription factor derived cell types in common media conditions, and using them in applications from programming organoids and cellular composition all the way to bioprinting large tissues composed of hundreds of millions of cells per ml of whatever cell type you can create with these pre-programmed cell lines. And I want to point out, one of the reasons we were able to do the bioprinting is that we've pre-programmed them into these iPSCs, which have this near infinite capacity for self-renewal and proliferation. So we can make large numbers of these pre-programmed iPSCs to the point where we can actually start making large amounts of ink that we can then do the bioprinting with. So the next steps so what are some of the next steps that we could take with this project? One of them is, what other organoid systems can we apply this concept of orthogonally induced differentiation to? We've demonstrated it in brain organoids, but it'd be also nice to see what other organoid systems is this system compatible with. Is there something unique about brain organoids, for instance, that makes it amenable to this? Or is the system generalizable to all differentiation protocols? And the second one is a question that came up earlier, is how do we create organ-specific cell types? Because we know we're using these transcription factors to derive these specific cell types, they're not necessarily organ-specific. For example, our endothelial cells are not specific to brain, they're just endothelial cells. And they don't express the typical markers that you would expect of brain microvascular endothelium, for instance. So how do we create these organ-specific cell types so that when we go into these organoid systems, we can then get more physiologically relevant cell types in them? So yeah, that brings us to the end of our presentation. And we'll take all sorts of questions now.
Yeah, thank you guys. Great presentation and really interesting work from beginning to end. Also great images. I mean, really beautiful. We do have a few questions just to kind of follow up on what you were just talking about with the ECs you said are not necessarily organ or tissue specific. Do you think that extended culture could maybe cause them to take on that kind of phenotype over time? What do you imagine would have to be adjusted?
I think so. One thing I think that's specific for brain is that we need the other cell types that the endothelium are sort of predisposed to interact with. And so Jeremy, when he was presenting, had a picture of the neurovascular unit where we had these endothelial cells that were then in contact with pericytes and astrocytes. And we don't have those cell types programmed into our organoids yet. And in the case of pericytes, they're not going to naturally arise in brain organ differentiation. In the case of astrocytes, they take months to develop natively in these organoid protocols. So for us, I think there is a possibility that these endothelial cells don't really know how to interact with neurons specifically, and so they're not getting the right signaling from the rest of the organoid to become more brain-like or brain microvascular-like. I'd be curious to see what happens if we cold culture these cells with like astrocytes or something. Right, we haven't done that yet.
Yeah, whether or not it's autocrine, paracrine, like it's implied, or in the future, a lot of people are working on even adding an additional cassette using an orthogonal stimulant to activate another set of transcription factor, maybe for maturation purposes, right? This is not just in endothelium, but in many other different cell types. So there are ways to not only engineer or potentially in co-culture, like Aric was describing.
Yeah, cool, awesome. And with that, how many cell types you guys feasibly think that you guys could be included in your bioprinting setup?
Reasonably or in theory?
Either. give me your what's like a moonshot we think.
So we could do it relatively comfortably with 3 different cell types. When we were doing the multi-material bioprinting, like having 3 different cell lines in culture wasn't too big of a challenge because they all took the same media. I think once you get to more than that, I think you're going to run into some issues with just the timing of getting all the cells aligned, and even the time it takes to print all of these cell types together. We already kind of showed that there is a time component to how fast you need to print in order to maintain good cell viability. So it's
might need multiple nozzles.
You might need multiple nozzles.
Right. Okay. A very clever way, and like maybe even in the future you could print one layer and then like encapsulate it and then print on top of it. Like introduce some kind of Z stack.
Yeah.
So there are probably clever ways to do it, and I think that's why it's showing that it is possible. Even 3, we've even in the Lewis lab is multi-material nozzles. So there is.
I think if you get past like 7 or 8, I think you have to really ask why I need this many cell types.
Right.
But I think it'd be possible to do around 7 to 8. At least with the technology we have that I know about.
Right. Again, kind of taking a little bit of a different direction. Just curious, you guys showed an intro slide in the very beginning about some of the other people that have been, a big part of this work. And there's, I think, a co-first author who's not with us today. What kind of role, how did they kind of aid in the conception? Do you guys want to just talk a little about them and their role a little bit?
Yeah. I did mention a little bit that the conversations about this work really stem from Mark's conversations with Alex Ng. So Alex Ng's transcription factor library paper in Nature Biotech, he was developing that years ago. And through one of our our center grants, it's called Center for Genomically Engineered Organs, that put like Jennifer Lewis in a room and George in a room and like Ting Wu in the room from Harvard and Ed Boyden. So there was a lot of like cross-discipline integrated collaboration, and this kind of like genesis of like, how do we utilize bioprinting with like engineered cells in like these like interesting like organoid space. So that's he really kind of was the genesis of that idea with Alex Ng. And then I actually was a first year grad student hearing these conversations, like, I want to come into this interaction, seeing the luminaries talking about their technologies and how they mesh very well. So he is really, he and Alex are the genesis of this idea.
Awesome. Well, I mean, this is a great time chat. We have these both presenters. Presented some awesome work. If you guys have any questions, please send them through. We just want to give you guys a couple minutes to continue to give these guys, to grill these guys a little bit. While we're waiting for, what was the most exciting or enjoyable part of this project for you guys? If you could just select one moment where you're just like, oh man, this is just awesome.
I think for me, so a little bit of background about myself. So my undergraduate degree wasn't in biology at all. Like I did my undergrad in electrical engineering. And so I never really thought about how we can start using something like 3D printing and do all sorts of biological experiments, or like I didn't even really know what a cell was coming out of undergrad. And then starting grad school, it's like, oh wait, now I actually have to try and keep something alive for my PhD. And I think just the overall experience of like making that transition, working with people like Mark and Jeremy, who helped make that transition, was a really exciting time in my life. It was also very stressful. I'm not gonna lie. It was a lot of hard work, but I think just having the opportunity to even think about some of these problems and trying to learn a whole new discipline is really kind of what was exciting for me.
I think it's kind of a excitement, but also, well, I mean, I loved working with the guys in the trenches. That's for sure. Sometimes you need all three of us to do something, but I remember getting the single cell sequencing data, or having to prep the library, and there was a humongous snowstorm, and I had to rush to get this all in before Harvard closed. And then I had to trudge back. I was the only one walking through from Longwood, the medical area, to the main campus near where I lived, and just having that sense was like, you know, we were toughing it through. It's like a good metaphor of this process. I was the only one. It was whiteout. Like I felt like I was gonna die out there, but the things we do for science, right? You gotta get it in. You gotta get the libraries in.
Yeah.
You know what I mean? And I nearly died that
100%.
night, but we got that. We got it done. Very memorable. Maybe not necessarily exciting, but you know.
Memorable. Good. So we do actually have one question from the chat. They said fantastic work, and one of the questions they had was, have you observed any contraction of the bioprinted constructs along the time length of culture?
I think probably it'd be easiest to go back to this this one. So if you look at the day 4 condition with the wild type iPSCs, we do see a little bit of contraction where the cells are, the filament has gotten a little bit thinner relative to the other conditions. You see places where the filament started breaking because you've had these cells that kind of pulled apart. So you do see a little bit of contraction. And one thing that you don't see in these images is you also see ECM degradation. The jubbin layer that we've cast on top of these tissues, the iPSCs, and particularly the endothelium, will just degrade it and start breaking apart the gel. So you do see a lot of contraction, and you do see a lot of ECM degradation in the process.
Thank you. Another question we had, out of curiosity, just how many flasks did you need to get to get 1 ml of cell bio ink? They also congratulate you on the hard work.
A lot of flasks.
Okay. So we could get 50 microliters from a T225. So I think that means you need 20 T225s to get 1 milliliter of ink.
You got stacks on stacks.
Yeah, those incubators were full.
Yeah. So luckily all of these prints didn't require that much ink, but if I had to make a milliliter of iPSC based ink, somebody at Stem Cell Technologies is going to have a very good day. For all the media we would need.
We've definitely used a lot of empties. Sometimes they would need me to, I was at Church Lab, they were like, we need 2 bottles now. So like I would get into Uber like with like 2 bottles and just get hand delivered to them.
Yeah, at the peak we were going through over a bottle of stem cell media a day. Yeah, for this bottle.
Oh my goodness.
Yeah.
Oh wow. And you guys were using empties or plus?
This was done in empties, or we were still a little bit worried about empties or plus at the time. So all in empties, so every day it was just a bottle of media, just out of the fridge and into the flasks.
The chat also wanted to congratulate you on the hard work and just wanted to say I can't imagine the amount of time that it takes just to culture all these cells, which you guys just spoke to. I mean, I'm sure that just had to be a lot. I tell you what, if you guys have a picture at all of just stacks inside of an incubator, you can send it to us and we'll tweet that on the paper trail just so that people can kind of see the amount of effort that goes into printing some of these beautiful images you guys have here.
I don't think I have a picture of all of the flasks. I do think I might have a picture somewhere of all of the organoids we cultured for this project, and there's just like plates on plates on plates of organoids all running in parallel. I'll have to find that in my, it's probably somewhere on my phone, but yeah.
I've just been like silently chuckling back here, just listening to you guys chat. Yeah. Thank you guys so much for all of your answers and for that really great presentation. It's just about 4 o'clock, so we're gonna wrap up. So I just want to thank everyone who came to our stream. Thank you for coming. Please come back again. We have another speaker towards the end of the month, so check out all of our website and our Twitter. We'll post that when that's up, and then this presentation will also be available on our YouTube probably by the end of the week or next week. So yeah, just check out all of the different things that we have to offer. Thank you guys so much for presenting. And yeah, I'm gonna end the stream.
Bye.
Bye.
Bye.